welcome to the latest Spotlight On interview. Today I'm joined by Andrew Sharrocks, and we're going to be talking about cancer epigenetics. Andy, if you could please introduce yourself and tell everyone a little bit about what you do.
Okay, so I'm Andy Sharrocks. I'm a professor of molecular biology at the University of Manchester. So my lab works on gene regulation, particularly how cell signaling impacts on gene regulation. Also on chromatin structure, how that impacts on gene regulation. So in the past, we've worked on a variety of systems, ranging from yeast through other systems like stem cells. And recently we've moved to studying cancer, in particular esophageal adenocarcinoma and the gene regulatory networks that are at play in that particular tumor type.
So why investigate the epigenetics of esophageal adenocarcinoma in particular? How does this disease differ to other forms of cancer?
Okay, so esophageal adenocarcinoma is a particularly deadly disease, so there's high incidence and very poor survival. So part of this is because we don't understand the molecular pathways involved as much as we do in many other cancers. So one of the issues with esophageal adenocarcinoma is a highly mutated cancer type, and yet there are no recurrent mutations. So P53 is highly commonly mutated, as in many cancers, but then the next highest incidence is probably something like the receptor tyrosine kinase, RB2, at 20 to 30%, and after that, not very much. So the usual common drivers aren't there in this cancer. So things like BRAF in melanoma, APC in colon cancer, or oncogenic fusions in leukemia, they're just not there. So getting targeted treatments becomes difficult. At the pathway level, you can see things slightly differently. So things like the receptor tyrosine kinase pathways, if you take different components and look at mutations within that, then you see much higher prevalence. So maybe 60–65% of all tumors would have that. But again, the common DNA mutations just aren't there.
So focusing on epigenetics specifically, what insights and advantages could studying epigenetics have in our understanding of this cancer and other cancers?
Okay, so as I just explained, there are very few common DNA mutations. The next obvious thing to look at is epigenetics. So the DNA is not naked in the cell, it's encased in chromatin, and chromatin controls the availability of regulatory elements and control gene expression. So equally, epigenetic changes may cause a change in cell phenotype, which gives you the cancer phenotype. The other thing about epigenetics, it can also give you an idea of where the cells come from in the first place, so your cell of origin of cancers. So that's something we try and pursue in our research, trying to understand, the basic wiring of cancer cells and where that rewiring originates in the first place.
So you spoke a little bit about chromatin. So what is the importance of chromatin unraveling, and how do you go about investigating such a dynamic process that's constantly changing? What are some of the challenges in exploring a process that's changing?
Okay, so as I just explained in the previous answer, really, that the DNA is encapsulated in chromatin. You need to then remodel the chromatin to get changes in gene expression. So you're revealing enhancer elements, promoter elements, and other regulatory elements in the DNA to allow access to the machinery that turns on gene expression. Equally, you can do the opposite. You can close down chromatin, and closing down chromatin will then shut down gene expression. And so that would be in particular tumor suppressors, which are usually stopping tumorigenesis. So you're then shutting those down, allowing cancer to occur, as opposed to opening up things tends to be oncogenic processes and oncogenic proteins which are produced from that. You did mention dynamics, so studying dynamics is always very difficult. Chromatin is actually not that dynamic compared to some other processes in the cell. The dynamics of chromatin occur in hours and days and weeks and months. So studying the dynamics is easier than what it is for some other processes.
Okay, thank you. So you were speaking about sort of the the genome level, the epigenome level, and then obviously the transcriptome level. So what do you think integrating multiomics and, this is a very rapidly developing field, multiomics, how do you see recent advances in multiomics, data integration, spatial? How do you find that in your research and just generally advancing our understanding of cancer and human health and disease?
Okay, so I think the clue is in the name with the multi-part of things. So if you want to understand any biological process, if you look at multiple facets of the same cell, you're able to then uncover deeper insights into what's going on. So that's where multiomics comes in. You're able to look at different aspects. So for example, there's chromatin level and there's a gene expression level. You can integrate those together to understand how the chromatin changes lead to gene expression. There's a protein level downstream. Obviously, that's what actually gives your cell your ultimate phenotype, to understand how that comes from gene expression profiles. So that's me talking as somebody who understands gene expression. But equally, if you want to phenotype cells, you want to get diagnostics, it can come from any particular molecular aspect or a combination of them. You mentioned spatial technologies there, and that's particularly important in cancer, for example, where you have a heterogeneous set of cells all interacting in space, where you have to understand where they are, how they interact with each other, and then the different types of cell types which are in there. Again, multiomics can give insights into that, combined with the spatial technologies which are now arising.
So you mentioned stem cells. So you also investigate how stem cells differentiate and some of the transcriptional drivers involved in this process. Could you walk us through your work in this area and how insights into this process is important for your research? So the transcriptional side as well as the epigenetics.
Okay, so if you think about stem cells, they're very primitive cells. They then go down through a series of steps and differentiate into a terminally differentiated cell. So our work in stem cells have been trying to understand those transitions, how you go from one cell type to another. So typically in the past, people have looked at the start point, your stem cell, and the end point, your differentiated cell, and you try and then work out how you got from A to B by understanding the transcriptomes in one, the transcriptome in the other, the chromatin state. What we've been looking is the intermediates in between. So when you mentioned dynamics before, that's where we look at what happens to the chromatin, the gene expression profiles, as you transition from a primitive embryonic stem cell through to a more differentiated state. The beauty of studying that system is you can do this in a synchronous manner. So you can start off with a synchronous population, you can then watch them through time as you go to the end point. So by doing that, we've been able to uncover transcription factors which define the two endpoints, but also the central transition point. So in mouse embryonic stem cells, for example, we identify ZIC3 as a transcription factor, which is only important in the transition state, but not in either of the two end states. So if you then go back to where this relates to cancer, if you think about cancer, it's the opposite. So cancer cells are effectively an undifferentiated cell, which have come from some differentiated, a partially differentiated cell in the adult. So it's a process in reverse, effectively. So studying that is effectively the same, but we're looking at how you go from a differentiated cell potentially to an undifferentiated cell. And then that process then goes one step further, where the cancer cells change properties to a metastatic state, which again you can see again as a progression from one cell state to another. So the process is pretty analogous.
So obviously these transitions happen over time, and recent advances in temporal analysis have allowed researchers to track and analyze these biological events as they occur over time. How do you see these new technologies and advances helping your work, as well as other investigations into how stem cells differentiate and how cancer cells develop?
Okay, so I mentioned how important looking at things temporally is in understanding how things go from A to B into this intermediate state. And especially if you're looking at signaling events, and mentioned in esophageal cancer, for example, we have the receptor tyrosine kinase signaling pathways upregulated, and they're obviously signaling through to chromatin and gene expression. One of the problems with looking at signaling is it's dynamic, and again looking at endpoints, start points. So there are new technologies like LiveSeq, which have recently come out. We can look at gene expression changes in real time. So you're able to extract part of the cytoplasm from individual cells and analyze that. So it's quite an exciting technology because you're able to sample the same cell multiple times. At the moment, most single cell technologies are not able to do that, and we have to take snapshots. You can do that through multiomics, so that's fine. You can connect things together through a multiomics on single cells. But I think the new technologies where you're able to look at what's going on in a single cell over time are particularly exciting. Can't do that with chromatin changes at the moment. I can't envisage a way of doing that at the moment, but if we could do that, again, it would be really exciting.
Thank you. So going back to epigenetics and esophageal adenocarcinoma, do you anticipate epigenetic insights into this cancer and other cancers translating to patients in the future? Will these studies help cancer patients in the clinic in the near future, or do you anticipate that sort of being a far off, distant future?
Well, there's multiple answers to that question. So what epigenetics can give you is an insight into tumours which other types of analysis don't particularly give you. So transcriptomics, DNA mutation analysis, typical genomics can give you one answer. Epigenomics can reveal new things. So in a way, that's giving you new pathways, new therapeutic targets potentially, potentially new diagnostic targets. So there are techniques which are coming out where you can look at circulating tumor DNA, for example, from patients, and you can then look at the fragmentation patterns, and from that you can infer the chromatin state of the patient. So from that, you can then begin to understand the pathways which are changed in patients. So as a diagnostic tool, you can say that the patients have cancer, so it's a non-invasive biologic tool from blood, and B, you can then begin to understand what those changes might be. So yes, there are potential future uses of epigenetics in revealing new things, but there are also potentially new diagnostic approaches through non-invasive blood sampling.
Like liquid biopsy.
Yeah, liquid biopsies, yeah, indeed.
So do you think these epigenetic insights could also, you mentioned diagnostics, but do you think it could also be used to inform precision medicine treatments?
Again, that's possible. So it gives you effectively the same. So one of the beauties of doing epigenetics and the sort that we do is we use open chromatin accessibility assays. What that does is reveals not just individual genes, but also programs. So if you get a transcription regulatory program regulated by a particular transcription factor, it gives you an insight there to immediately go in and intervene in that pathway. Obviously, targeting transcription factors isn't as easy as signaling pathways, but people are doing it. So there are ways of doing it. You just have to think a little bit out of the box to be able to do it.
Okay, thank you. So just to finish with the interview, what novel research coming out of your lab are you most excited for? What should we be keeping an eye out for in the near future?
Okay, so something we're doing at the moment, which is something I won't talk about necessarily in the conference, is we're looking at transcriptional repression in cancer. So that's a less studied element of cancer, probably because it's just more difficult to study. So we're looking at this in different ways. But one of the things we started to look at is the role of phase separation in transcriptional repression, and then applying that to cancer. So at the moment, we're currently not doing this in cancer cells, but working up methodology to look at repression and how phase separation might do that. And in the future, this could be a completely novel way of treating cancer patients, rather than trying to affect binary molecular interactions. If you can split apart phase-separated condensates, which are involved in key processes in cancer cells, then that could be a new therapeutic angle and may open up the field for new therapies.
That's really interesting. I haven't heard much about that at all. So could you explain in a little bit more detail what phase separation is?
Okay, that'll be so. So normally you think of proteins and different molecules interacting through the lock and key hypothesis, where you've got one thing binding to another, and then another, another, another. You build up complexes, you build up assemblies, etc. Phase separation is something slightly different, where you have multiple low affinity interactions, which bring things together and hold it in a more liquid environment. So it's not a solid, but it's more of a continuum of things. So that's driven by low affinity interactions as opposed to the high affinity lock and key hypothesis. So that's driving compartmentalization of various things in the cell. So you get various granules, the nucleolus is another example of that, but also at a smaller level in enhancer regions. So put all the transcriptional regulatory apparatus in there. And then what I was talking about there is similarly in repression, where you're shutting down gene expression, you bring things together in these localized conglomerates, these phase-separated areas, to control gene expression.
Did you sort of anticipate something like AlphaFold or other proteomics tools coming into play for that sort of field of research?
Yeah, probably not. So the issue with AlphaFold is it's dealing with globular domains. What AlphaFold can't deal with at the moment is the non-folded domains. We don't know what the structures are, and these non-structured domains are what are driving phase separation. So this needs new insights into what these disordered regions are doing in proteins, which can't be readily predicted through just comparing across different structures, which is basically what AlphaFold does. Is it takes one structure and looks for something which may map onto that structure. Right. So it's the antithesis to AlphaFold in many ways. It's the new frontier to understanding what these other bits in proteins do, which we've conveniently ignored over the last few decades.
Right, because they're difficult to look into.
Yeah.
Okay, thank you. That's really interesting. So I just wanted to finish off with some questions about Triomics. So you're going to be speaking at the Triomic Summit in London. Why are you looking forward to the Triomic Summit? What do you think people could get from visiting the Triomic Summit?
Okay, so for me, there are the three strands there, just sort of map onto where science is going and where my lab's going at the moment. So you've got the multiomics side of it. So we discussed that in this interview quite a bit. And multiomics is important in understanding biological processes. There's the spatial technologies. These are just increasing at a pace now. So keeping up to date with what those are, how people are applying them is really important. So we can do single cell work in when you disaggregate tissues. It's really important to understand how things come back together in situ, especially in cancer, where you've got these really complex environments. That's something we now integrate into our workflows. And the other element, genomics of cancer, obviously that's the other omic which people have looked at extensively in the past. So we have to build on these more functional omic methodologies on top of that. So coming to the conference is going to give us an idea of all those three things. Unfortunately, I can't be in two places at once, so the two strands I may have to catch up on one of them after the conference.
Yeah, thank you. So what will attendees look forward to learning from your talk specifically?
So the talk's going to have two different elements to it. The first part is going to be really about understanding the basis to cancer and where it comes from. So I mentioned before about development going in reverse. So I'll present some data which essentially is taking epigenetic data of open chromatin accessibility, where we're able to then cross-compare what happens in tumor evolution to human embryonic development. So I'll show you how the two things interrelate and how tumorigenesis looks like it's going backwards into the embryo again. So we're going back to a primitive state. The second part, I'll explain how you can go from this accessible chromatin opening to give you insights into tumor biology. So integrating RNA-seq data on top of that to understand how regulatory elements work, and how that then tells you new things about what goes on in esophageal cancer.
Thank you very much.